שלום לכולם, אני רק מזכירה שחלק גדול מצטרפים אלינו בזום, אז לא רק... אני אומרת שחלק גדול מצטרפים אלינו בזום, כך שהנוכחים שכאן הם רק חלק קטן מהנוכחים. אז אני שמחה לפתוח את טקס הענקת פרס משפחת יודעים למצוינות בחקר הסרטן. יש לנו היום 3 זוכים בפרס. אני קודם אפתח בכמה מילים על פרופסור מוסא יודעים ובברכות, וכמובן שאחר כך זוכי הפרס כבדו אותנו בהרצאות על נושא המחקר שלהם. פרופסור מוסא יודעים נולד בטהרן בעיצומה של מלחמת העולם השנייה. אחרי שהוא סיים את חוק לימודיו באנגליה, הוא התקבל ללימודי רפואה באוניברסיטת מגיל במונטריאול שבקנדה, אבל בסופו של דבר מסיבות אישיות החליט לעבור ללימודי התואר השלישי בנוירוכימיה ולהמשיך את המשך לימודיו ומחקריו בעתיד לחקר הפרמקולוגיה של המוח. אני חושבת שהעולם הרוויח בגדול מהבחירה הזאת. לאחר סיום לימודיו לתואר PhD במחלקה הפסיכיאטרית באוניברסיטת מגיל, זכה דוקטור יודעים במלגת פוסט-דוקטורט והחל בסדרה של השתלמויות באוניברסיטת לונדון, קיימברידג' ודה פרנס בצרפת. ב-1975 הוא ביקר בישראל לרגל חנוכת בית הספר לרפואה בטכניון, ולמעשה הביקור הזה שינה את המהלך אחרי שפרופסור דוד ארליך, דיקן בית הספר לרפואה דאז, הציע לו להקים ולעמוד בראש המחלקה לפרמקולוגיה, שבעצם עמד בראשה עד 1994. הוא היה שותף לגילוי החלוצי של התועלת הקלינית של תרכובת הנקראת סלג'לין בטיפול במחלת הפרקינסון, מה שהוביל בהמשך לפיתוח תרופה לפרקינסון בעלת יכולות הגנה על תאי עצב, שידועה גם בשם המסחרי אזילקט. היא מוכרת כתרופה בסל הבריאות, משווקת בכל העולם. פרופסור יודעים הוא מחלוצי חקר תפקודי הברזל במוח, במצבי חסר או עודף, בתהליכי ניוון מוחי, במחלות פרקינסון ואלצהיימר. הוא היה אחד מהחוקרים הראשונים שפיתחו תרופות קולטות ברזל ויישמו את הגישות המחקריות החדשניות של תרופות רב-תפקודיות לטיפול במחלות המוח, ותרופות אלה מהוות את עוגן הפעילות של החממה לפיתוח התרופות שהקים לאחרונה. עם פרישתו מניהול המחלקה לפרמקולוגיה בפקולטה בטכניון ב-1994 הוא הקים וניהל את מרכז המצוינות החדש לחקר מחלות ניווניות של המוח, שהיה מבין הראשונים בעולם בשימוש של שיטות חדישות במודלים פרה-קליניים וגנטיים. מודלים אלה משמשים לאיתור חלבונים הפועלים כסמנים ביולוגיים לאירועים המולקולריים המורכבים שגורמים לניוון מוחי במחלת הפרקינסון. בנוסף להישגיו המדעיים הבולטים של פרופסור יודעים, הוא משמש גם בתפקיד מנחה ויועץ לסטודנטים, מדענים וחוקרים בארץ ובחו"ל. מחקריו פורסמו בלמעלה מ-900 מאמרים מדעיים וזיכו אותו בפרסים באותות כבוד רבים, בין היתר פרס אמת לשנת 2010, הישגיו בתחום מדעי העצב וחקר מחלות הפרקינסון ואלצהיימר, שהובילו לפיתוח תרופות למחלות קשות אלה בישראל. ולאחרונה, ביום העצמאות האחרון, פרס ישראל בתחום חקר מדעי החיים, שבאמת לכבוד הוא לנו שקיבלת אותו. פרופסור יודעים מחזיק במינוי פרופסור מחקר מדעי ופרופסור כבוד באוניברסיטת הונג קונג, שנגחאי בסין, באקדמיה הגרמנית למדעים לאופודיניה. הוא מייסד שותף של מספר חברות לפיתוח תרופות, ועם סיום הקריירה האקדמית שלו החליט להקדיש את השתת זמנו מרצו וגם ממון מכובד להקמת מרכז ישראלי למימון פיתוח תרופות בשלב מוקדם. הוא עומד כיום בראש חברת יודעים פרמסוטיקלס בע"מ, המגשרת בין השלבים המוקדמים בגילוי תרופות במוסדות האקדמיים לשלבים שבהם קרנות הון סיכון וחברות התרופות מוכנות להשקיע בתרופה חדשה. הוא נשוי לפרומה, מומחית בעלת שם להוראת ילדים עם לקויי למידה ואב ל3 ילדים. אז שוב תודה רבה ולכבוד הוא לנו, ואני רוצה להזמין את פרופסור אלברט טל.
שלום לכולם. מוסא יודעים, משפחת יודעים היקרה, הזוכים היקרים וקהל נכבד, אני שמח לברך את כולכם לכבוד כנס פרס משפחת יודעים בחקר הסרטן. זו השנה השביעית בו אנו גאים ומתכבדים להעניק את פרס משפחת יודעים לחקר הסרטן. עד כה הוענקו הפרסים ל8 זוכים, והיום מצטרפים לרשימה המכובדת זאת 3 זוכים נוספים. פרס החוקר לפרופסור רביד שטרוסמן ממכון ויצמן, והשנה התווספו 2 פרסים במסלול חדש, פרסים לסטודנטים המצטיינים לחקר הסרטן, למר נמרוד רפפורט מאוניברסיטת תל אביב, והשני למר גלעד גולדהירש מהטכניון. אני חושב שהנושא הזה, שמעבר לזה שאנחנו תומכים בחוקרים, אבל גם כן בונים את דור העתיד, זה אחד הדברים המשמעותיים ביותר שכולנו מחויבים לנו. יש לנו ברמב"ם קמפוס ייחודי, הכולל את הפקולטה לרפואה של רפפורט בטכניון, המאפשר שילוב של טיפול רפואי מתקדם מעולה, מחקר מדעי, טכנולוגיה וחינוך, ורמב"ם ממשיך להתקדם בשנים האחרונות. אנחנו רואים פה בתוך הקמפוס שהמכון האונקולוגי של יוסף פישמן, שאנחנו יושבים פה בבניין, בית החולים אייל עופר ללב ובית חולים רות רפפורט לילדים, הם תוספות חשובות למתקני הטיפול בחולים שלנו. במרכז הקמפוס מגדל התגליות הביו-רפואיות על שם אמסלי, אשר יהווה תוספת יוצאת דופן למחקר המתמשך במכון המחקר הקליני ברמב"ם. אבל חשוב הוא שהאנשים שאנחנו משקיעים בהם, אנו תומכים ברופאים, מדענים, מצטיינים בתוכניות השונות, אנו משקיעים בעתיד הרפואה באמצעות מדע בסיסי ויישומי, ואנו גאים להמשיך להוביל בתחום המדע והרפואה. אני רוצה להודות לפרופסור מוסא יודעים ולמשפחה על תרומתם העצומה והחשובה למדע בישראל, תרומתכם החשובה במהלך השנים, אשר מטרתה לתמוך בהישגים בתחום חקר הסרטן, תמשיך לשנות את עתיד הרפואה בישראל ותשפיע באופן ישיר על המטופלים, וזה מה שאנחנו עושים פה. פרופסור מוסא יודעים הוא מהאנשים הנדירים שמהווים דוגמה אישית לחשיבות המחקר למען הרפואה בישראל, וכאות להערכה ולהכרה זו זכה פרופסור יודעים, כמו שנאמר כבר מקודם, בפרס ישראל לשנת 2022, על תרומתו החשובה לקידום הרפואה והמחקר בישראל. אין לי ספק כי זכייתך בפרס מדגישה את הישגיך המיוחדים ואת תרומתך החשובה לחברה הישראלית, לרפואה ולמחקר לאורך שנים, ואין ראוי ומתאים ממך לקבל את הפרס המכובד והיוקרתי הזה. זכות גדולה נפלה בחלקנו שבחרת אתה ובני משפחתך ברמב"ם להיות הבית שלך שמעניק פרס חשוב כל כך לחוקרים בישראל. בשמנו ובשם כל משפחת רמב"ם אנו מבקשים להודות לך ולכל המשפחה על עמידתכם הנאמנה המתמדת לצידנו ועל תרומתכם יוצאת הדופן ורבת השנים לרמב"ם בפרט ולמחקר ולרפואה בישראל בכלל. ואתם לזוכים הנכבדים, אין לי ספק כי נמצאתם ראויים לקבל פרס חשוב זה ותעשו בו שימוש מושכל לקידום המחקר בישראל. אני מאחל לכולנו כינוס פורה ומהנה.
רוצה להזמין את פרופסור הקריה.
תודה לפרופסור בן-אהרון, פרופסור יודעים, פורמה, משפחת יודעים, כאן הרחבה, חברים, פרופסור אלברטה, פרופסור אירית בן-אהרון. אני ממש מתרגש פעם שביעית כבר בהענקת הפרס הזה, הייתי בכל התהליך של הבחירה עוד מהפרס הראשון, ואני זוכר טוב מאוד איך דנו עם פרופסור יודעים, מהו הדרך שבו נשים בחזית את המחקר בתחומי מחלות הסרטן בישראל. ואני חושב שבמשך 7 השנים האלה ראינו את התחרות העזה בין כל החוקרי סרטן המצוינים בישראל, ויש בישראל מחקר מצוין בסרטן, ובהזדמנות זאת כמובן אני מברך את הזוכים כאן, שבפירוש התחרות לא הייתה קלה. היו הרבה אנשים מועמדים שהיה צריך לבחור ביניהם, ואנחנו תכף נשמע מהזוכה את ההישגים ואת הכיוונים מחלות הסרטן וכמה חידושים מאוד מעניינים בתחום הזה. בהזדמנות זאת אני גם רוצה לברך מעבר לתודה את פרופסור יודעים על קבלת פרס ישראל, שהוא כל כך ראוי, ובשנה הזאת במיוחד אני חושב שזה מציין גם כן ציון דרך חשוב בהתפתחות של המחקר בישראל בתחומים השונים. במקרה של פרופסור יודעים זה בעיקר המחלות הנאורוגנרטיביות של המוח, אבל ההסתכלות כמו שאתם רואים היא הרבה יותר רחבה, לא רק נאורוגנרציה של המוח, גם סרטן, ואין לי ספק שהעשור שלפנינו אנחנו נראה מהפכה בטיפולים בסרטן, לא רק תרופתית, גם נכנס כאן כל הנושא של AI, שיטות חדשות, גם לזיהוי מוקדם, גם לטיפול מדויק יותר, אישי, בחולי הסרטן, גם לניבוי תרופות, ניבוי תגובה לתרופות, בצורה יותר מדויקת, כך שהטיפול יהיה יותר מדויק. הדברים האלה, אין לי ספק, אני רואה המון פיתוחים בדברים האלה, אין לי ספק שתהיה מהפכה ב10 שנים הקרובות, ואחת המחלות הכי קשות שעוד לא כבשנו אותה, וצריך לכבוש אותה. אז תודה רבה, ושיהיה כינוס מרתק. תודה.
רוצה להזמין את פרופסור יודעים.
Shalom Kulam. First of all, I'd like to thank all of you for the kind words you said. Maybe we forgot to say that he's also modest. I hope so. First of all, I'd like to congratulate the prize winners. I think we have done a wonderful choice. After 2 years of Corona, this is the first one of 2 years. I'm glad that we can continue our progress in giving prizes for really the best work done on cancer. My own work actually started from what I learned from cancer, because the neurodegenerative diseases, the aspect is completely opposite, face opposite to what happens in cancer. And many of the work that I did, really I went to even to cancer symposiums and conferences to learn what's the molecular mechanism of why in cancer the cells regenerate and proliferate, whereas in neurodegenerative diseases, the cells die. And we would like to regenerate the neurons that are responsible for causing Parkinson's disease and Alzheimer's disease. So we learned a lot from there. And through my life, I've been very lucky. I'm really honored that I received the Israel Prize this year, thanks to many people who recommended my name. And once again, I want to thank my organizing committee. I think you've done a wonderful job. I've eliminated myself from the organizing committee not to be biased. And they've done a wonderful job every year. They have chosen some of the best people who are doing really cancer in Israel. Thank you again, and I'm sure we're going to have a wonderful symposium today with the winners. Thank you.
אני רוצה להזמין את פרופסור יודעים לחזור אלינו לבמה יחד עם פרופסור ביאר ופרופסור אלברטל, למתן התעודות לזוכים. אז פרופסור רבית שטראוסמן. שאנחנו נשמע את הרצאתו עוד מעט. את נמרוד רפופורט, ואת גלעד גולדהירש.
אנחנו באקדמיה, צריכים לעשות evidence based.
עכשיו הגענו לחלק המדעי. פרס משפחת יודעים למצוינות בחקר הסרטן לשנת 2021 מוענק לפרופסור, 2022, מוענק לפרופסור רביד שטראוסמן על תגליותיו במחקריו פורצי הדרך בתחום המייקרוביום של הגידול האנושי והשפעתו על עמידות לתרופות. פרופסור שטראוסמן הוא רופא חוקר שסיים את לימודי הרפואה באוניברסיטה העברית בשנת 2001 ותואר PhD ב-2005 גם כן באוניברסיטה העברית. המשיך לפוסט-דוקטורט במעבדה של פרופסור סידר ובמעבדה של טוד גולו בהרווארד. מ-2013 הוא חוקר בכיר במכון ויצמן למדע. הרבה מאוד פרסומים וכתבי עת מובילים ביותר, זכה בהרבה מאוד גרנטים, פרסים רבים לאורך כל לימודיו והקריירה שלו, גם הוא מאוד צנוע, ואנחנו מזמינים אותו לתת לנו הרצאה על Optimizing Cancer Precision Therapy by Studying and Targeting the Tumor Microenvironment, microbiome. בבקשה.
Yes. Perfect. So, thank you very much. Professor יודעים, Professor בן-אהרון, Professor אלברט, Professor ביאר, it's a really pleasure and honor to be here and get this prize. I'm really thankful for it. I think it's a great thing that you support the oncology field in Israel, which, as was said before, really needs a lot of push forward so we can do better with patients. So I'll try today to highlight, indeed, I think, what would be novel directions, how one can really rethink some parts of it, and how can we use novel ways to look at basic science, but also to try to translate it and bring it all the way to patients. I want to start by stating that there's an urgent need for better precision therapy in oncology. So we really think that in 2022, we are at the era of precision medicine. But the truth of the matter is that most of the patients are not there yet. So many of the patients, they still don't get as precise therapy as we want them to have. And there's a lot of guessing involved in spite of all the research that was done over the last few years. For example, if you look at all the sequencing technology that have come forward over the last few years, less than 10% of the patients actually benefit from these sequencing technologies. Just to give you one example of many, I can give hundreds at least, is a waterfall plot of patients that had a specific translocation, the email for ALK translocation, so very precise genetic aberration, and they've been treated with ALK inhibitor. As you can see on the right, some patients respond and some patients on the left do not respond. So they progress in spite of this very precise therapy. And even here, which is the most precise and most accurate case that we can get, we still don't really understand who of these patients is going to fall on the right and enjoy the treatment and who's going to fall on the left side. And why is it? Why is it so difficult for us to really understand how to predict if a patient is going to respond or not? There's many questions to that. Cancer is a very, complex disease, but one of the answers, the main answer is probably rooted in the complexity of the tumor microenvironment. So as you know, tumors, just like any organ, doesn't contain just cancer cells. So here you can see a breast cancer tumor from a woman. These are the cancer cells, but there are many other components in what we call the tumor. There are the cancer cells, there are immune cells, there are blood vessels, stromal cells, and many other components. And all these components probably affect the way patients respond to therapy. So to give you an example from one of my first studies, so here we looked at the effect of stromal cells on the response of cancer cells to therapy. So we took different cancer cells, treated them with drugs, and then added stromal cells and asked how can these stromal cells affect the way cancer cells respond to therapy. So if you look, you can see that they are, can you see my, yeah. So you can see these are the stromal cells in the whale. These are the cancer cells. As here you see there's not much crosstalk between the stromal cells and the cancer cells. But if you look at another stromal cell, another cancer cell, you immediately see that there's a very tight crosstalk between these cells. And we found, I won't show you here, hundreds of cases in which cancer cell, when you just treat them, they respond to therapy if they're alone. But if you add stromal cells together with them, they become resistant to therapy. Here's one example, and you can see a melanoma patient. This is a dermis, that's part of our skin. These are the cancer cells, the melanoma. And we found that the stromal cells, you can see here are brown, they secrete a molecule that's called AGF, peptide growth factor, and it can protect the cancer cells from a very common anti-cancer drug that we usually treat patients with. But looking at these hundreds of cases, I want to maybe zoom in on one case that we found particularly interesting. This was a case of a drug called gemcitabine that is very commonly given to cancer patients. And we looked at different cancer cells like RKO, that's a colorectal cancer, but we looked also at pancreatic cancer. What we found is that if there's no fibroblasts and you add gemcitabine, you can kill the cancer cells. But if you add the fibroblasts, and this was a line that we called the HDF, the cancer cells continue to proliferate. Also, if you just add in the preconditioning media that is secrete, so what's the secretions of the stromal cells, that's enough to protect the cancer cells from chemotherapy. And we asked how? How can stromal cells protect cancer cell line from chemotherapy? So we made a lot of research around it. It was very difficult to point it out, but eventually we found that it wasn't really the stromal cells, but actually the stromal cells that we were using were infected with bacteria, unique bacteria. It was the bacteria that rescued the cancer cells from chemotherapy, as you can see here. So the HDF, that's a PCR that we looked for the specific bacteria, and it was the bacteria, these HDF cells were positive for it. So we asked, are there really bacteria inside human tumors, right? Can bacteria in tumors protect cancer cells from chemotherapy? So Lior and Michal in my lab looked at biopsies from pancreatic cancer patients, from pancreatic tumors, and we found that indeed in the majority of pancreatic cancer patients, you can find bacteria inside their tumor. That was the first time that bacteria were described inside pancreatic cancer. And why do you care that there are bacteria? Oh, sorry. First of all, we also able to stain them. So you can see here in brown, you can see the bacteria inside pancreatic patients. You can see them by fish in different ways. And you can see in electron microscopy, here you can see this is everything you see here is one cell, human pancreatic cancer cell, that's a nucleus. And here you can see the bacteria sitting right in the cytoplasm next to the nuclear envelope. So why is it important? So for example, we found that these bacteria indeed can protect cancer cell tumors from chemotherapy. So we took mice and they had colorectal cancer tumors or pancreatic cancer tumors, and we injected bacteria into the tail vein, and these are unique bacteria that home to the tumors. What we found was that if you have bacteria, the tumors grow under chemotherapy, right? They're completely resistant to therapy. But if there's no bacteria, then you can control the tumor and they don't respond to chemotherapy. And they respond to chemotherapy now that there is no bacteria. And what's the mechanism? How can bacteria protect cancer cells from chemotherapy? So what we found was that usually when you add gemcitabine, this drug goes into the cell, goes into the nucleus, and then it incorporates into the DNA and causes DNA breaks, and that's the mechanism by which it kills the cancer cells. But what happens if you have bacteria? We demonstrated that when you have bacteria, the gemcitabine inside the cancer cell, instead of going into the nucleus, it's being actively pumped inside the bacteria by a transporter that we recognize as NAPC. And then we found that there is a specific bacterial gene that is called CDDL that can degrade the chemotherapy. So the bacteria basically cut the chemotherapy and inactivate it. So if you have a cancer cell and there is bacteria inside the cancer cell, it would become resistant to chemotherapy. And more papers came out in the last few years demonstrating that this is not just relevant to our mice models, but actually relevant to patients. For example, that's a very nice study from MGH, Mass General Hospital in Boston, and the Ludwig Maximilian University in Germany, they followed patients with pancreatic cancer, and usually, you know, this patient has a dismal prognosis, as you can see here. So most of them die. There's less than 20% of them that survive after 3 years. But if these patients, just by chance, were treated with quinolones, which is a specific antibiotic that target the bacteria that can degrade the drug, if these patients, for any other reason, got this antibiotic, then they would actually survive much better. And we're going to start very soon clinical trials in Israel, actually trying to demonstrate the same thing. And right now in my lab, there's different students that follow other projects that I won't go into right now. But what we do now, we really look at this angle in which we look at how different bacteria can affect the way cancer cells respond to a therapy. Aviva and Gili both following very fascinating stories like this. But then we ask the question, so we found that there are bacteria inside pancreatic cancer, but are there bacteria in every cancer type? Right? And are they the same bacteria? Are these bacteria unique to cancer, or do different cancer types have different bacteria in them? And what are the functions, other functions that these bacteria may have? How do they affect the way our patients respond to a chemotherapy? And Dvora Neumann and Ilana Leviatan from my lab, postdocs, and Gary and Noam, and many other students helped in this fairly large project. It was with many other collaborators, very generous collaborators from all over the world that helped us to get these samples and look at them. And this was published 2 years ago. And what we did here, we took altogether 1,500 tumors, human tumors, and hundreds of negative controls, because it's very tricky to look at the microbial component of tumors, because you always worry that it's contaminated for something, you know, from the environment. And we had 9 medical centers. We optimized multiple methods to really deeply and very accurately characterize the bacteria inside these tumors. What we found was that one can find bacteria almost in every tumor type. Not every patient has bacteria, but many of them. For example, here you look at almost 400 breast cancer patients, and you can see that some of them are negative for bacteria and some are highly positive for bacteria. This can be shown by PCR or by staining and by different methods. And if you look between cancer types, you actually find that different cancer types have different bacteria. So there is a cancer type specific signature of bacteria. So here, for example, you look at different cancer types, right? In pancreas, in bone, in lung, and so on. You look from the phyla level, so very high above, in different bacteria, but you immediately see that there's huge differences. Just even one example, proteobacteria, as you can see here, the blue are very common in pancreatic cancer, very rare in colon cancer. So in every cancer type, you find different bacteria. And if you look at these specific bacteria, you see that you can actually cluster and make a very distinct signature of bacteria in every cancer type. So what happens if you now compare the bacteria that are in the cancer to the normal tissue? So do normal tissue have bacteria, and are there somewhat related to the cancer? So it's really hard to get normal tissue, because usually people don't donate their normal organs, but we have what we call the normal adjacent to tumor. So when you take a tumor out, we usually take some normal tissue with it. So we look at it, and what we found was that in most cases, you can find the same bacteria, similar bacteria in the tissue around the tumor, and still when you compare the normal and tumor, you find that there are some bacteria that are distinctively present in cancer or enriched in the cancer or in the normal. And usually there are more bacteria in the tumor versus the normal. So here you can see, for example, that we found more bacteria in breast tumor versus the normal adjacent tissue versus normal breast. So the cancer tissue is enriched with bacteria as compared to the normal. So where are these bacteria present? Are they between the cells, in the blood vessels, in the immune cells? Where are they in the tumor? So we stained hundreds of tumors, in different types of tumor, and we came out with what we found was, maybe I also can see that you also see it. What we found was that most of these bacteria are intracellular. So they are found inside the cancer cells, not between them, but inside the cancer cells and sometimes inside the immune cells. Okay? As you can see here, for example, you can see it in electron microscopy. So that's one cell, that's the nucleus, that's a nucleolus. You can see here the bacteria with TEM, electron microscopy, and with anti-LPS immunofluorescence on the same cell. And most of the times you'll find the bacteria in a very specific location inside the cancer cells. As you can see here, they're located right next to the nuclear envelope for a reason we still do not understand really why. And sometimes they're also present in immune cells. That's a breast cancer patient. There's a lot of CD4, D5 cells, so a lot of immune cells infiltrating the tumor. And if you look at fish or in immunohistochemistry, you can find that many of these immune cells also have bacteria in them. So most of the time, the bacteria in the cancer cells and sometimes in the immune cells. Are these live bacteria? So maybe these bacteria that go to the tumor, but they're not live anymore. So we cultured bacteria for many patients, and again, we can culture live bacteria from these tumors and show that many of the bacteria, I don't know if all of them, but many of the bacteria in tumors are actually alive. So why is it that you have different bacteria types in different tumor types, right? Why is this connection between the type of tumor and different bacteria? We think this has to do with a different microenvironment that every tumor has and the specific functions that the bacteria has. And just to give you one example, so first of all, looking at the functions, we found that different tumors have bacteria with different functions, not just which bacteria are there, but what are the functions. And to give you one example, if you we looked at lung cancer tumors from smokers and non-smokers. And we found that there are different bacteria, but also different bacterial functions in tumors from smokers and non-smokers. What are the differences? If you look at what are the most highly enriched genes, you find that in smokers, the bacteria can degrade nicotine and toluene, acrylonitrile, and chlorobenzene. These are all smoke-related metabolites. So people that smoke, they have these metabolites in their tumors, and there are bacteria that probably can feed on these metabolites or use them for something in order to survive in these tumors. And do these bacteria affect the prognosis of the patients or the treatment? So we have lots of data. I'll just show you one example. So here we looked at melanoma patients that responded or did not respond to immunotherapy. You know, immunotherapy is the first line for most of melanoma patients. We can find that there are different bacteria in patients that respond and do not respond. And that make us think maybe these bacteria, different bacteria, they actually affect the way cancer patients respond to immunotherapy. To show also that the bacteria affect the local immune system, we had this nice paper last year together with Yardena Samuel and Jen Wargo, demonstrating that the bacteria that are intercellular, their proteins are presented on the cancer cell by the MHC class 1 and 2 in the tumor. So the immune system really sees these bacteria to some extent. And Yara from my lab and Stefan and now also Tatiana helping them, over the last few years, they carried out, Yara led it mostly, they carried out an interesting clinical trial. We followed 300 cancer patients that were about to start to get immunotherapy. And we took a biopsy from them and asked which bacteria they have in the tumors and are they going to respond or not respond to immunotherapy. And we found some bacteria that really highly enriched in responders or non-responders. And to test if this is just correlation, if there's a causal thing around it, we took these bacteria, we made them luciferase positive, so we can trace them in vivo after we inject them into mice. And we demonstrate, first of all, that they home only to the tumor. So you can see here, this is a tumor of a mouse before we injected the bacteria. And we inject the bacteria into the tail vein, you can only see them in the tumor. So these bacteria home specifically to the tumor. And in the tumor, different bacteria have different effect that is nicely correlated to what we had before. So for example, here you can see, we took 2 strains of bacteria, and one of them really suppressed the immune system in the tumor. This is a strain that we found to be enriched in non-responders. And one of them elevates the number of CD8 or CD4 or macrophages in the tumor. So the bacteria can really affect the immune landscape and the response to therapy. So that make us think maybe we can actually do something with it to treat patients. So I want to give you one example of things that started in the bench and we're trying to take all the way to patients. So a group in my lab and now in a company that I'll tell you about in a second, led by Oded Sandler and Dvorah Neumann and Reut and Tanel and others, they take these specific bacteria that we found to make patients respond more to immunotherapy. And what we want to do is we want to inject them into patients because they only home to tumors and they make the patient respond. So just a few months ago, we started this company vaccine, which stands for bacterial vaccine. What we do there is the following. We take these unique bacteria that we know that are correlated with response to therapy. First of all, we edit their genome because we want to increase their safety to make sure that there's no safety issues. But there's two more tricks that we do that are highly valuable, I think. One, we make the bacteria to present the tumor neoantigens. So when the immune system, when the macrophages, dendritic cells, take in the bacteria and process them, as usually they do, what they actually see is not just the bacteria, they see the neoantigen of the tumor of the patient. And what you get is a T-cell clone and antibodies that are directed against the patient's tumor. And the second thing we do is we make the bacteria secrete basically all the immune payloads that one can wish. Usually it's very hard to treat these patients with immune cytokines that have a high systemic effect. But here we have these bacteria that once injected IV just go only to the tumor and they can secrete there as a cytokine, any cytokine you want to recruit T cells, to make them active, to recruit macrophages, to change M2 to M1, everything you want, you wish, you can actually put on these bacteria. And we're really advancing very fast, hoping to reach patients very soon. And these are experiments in mice in which you can see that you take mice models that are not responsive that much to immunotherapy or any other therapy actually. And just by injecting bacteria to the tail vein, the bacteria directly go to the tumor and we completely cure these mice. And hopefully we'll be in patients soon. I want also to say that not only bacteria are found inside tumors, we also find that fungi are present in tumors. This is from a human colon, and you know there are many fungi in the colon. So we asked, are there also in the tumor? There's hundreds of thousands, millions of fungal strains in the world, and many of them were also, hundreds of them were discovered in patients. So we asked, are there fungi also in tumors? So this was a work that is now under revision, a third revision already, hopefully will be published soon. It was led by Lian Narunsky and Ilana Leviatan, in collaboration with Sahi Pilpel, and together also with Rob Knight, his student Greg, and many others. And what we did here, we profiled more than 17,000 tumors to the presence of fungi. For the sake of time, I didn't bring you the results. I'll just tell you in one sentence that the answer is yes, we found fungi in most. I'm sorry? Take it down. Perfect. Perfect. Innovation. So yes, the answer is, sorry? So yeah, so what we found, I won't show you the results, but we looked at 35 different cancer types, more than 17,000 tumors. One can find really fungi in most of these tumors. And I think more interestingly, I didn't bring the results. The presence of fungi is really tightly correlated to response and overall survival of patients, even more than bacteria to some extent. Interesting to think about why. You can speak about it some other time. I think the last thing is to say in this part, and then I have another short part, is that, you know, the hallmark of cancer is a big review that comes out now for the third time every 3 years, and polymorphic microbiome, including the tumor microbiome, now become a hallmark of cancer. So something that was hardly known a few years ago now become a hallmark of cancer. I think it's really an exploding area that is really interesting and will have a really great effect on patients. But we started with the idea that there's many different things in the tumor microenvironment, right? There are stromal cells and immune cells, and I showed you bacteria, I showed you fungi. Obviously, there's many other things that we don't understand. So it's actually interesting. It's actually easy to understand why is it so hard for us to really predict what's the best therapy for each patient, because it's a really highly complex environment that we don't fully understand. So what can we do now, not in 30 years, in 100 years when we understand everything? How can we help patients now if they want to better choose their therapy? So there's many ways to go about it, but I want to show you one solution that we came out with. This was a project led by Nancy Gabbert and Odette Sander from my lab, and it was just recently published. So the idea was here, maybe we shouldn't be so smart, maybe we just functionally check it. The idea was, can we take a tumor from a patient and bring it to the lab, and instead of dissociating it into single cells, can we just treat the tumor as is with drugs and see if it's responsive or not when it contains everything that originally is there. So we started this company called QResponse a few years ago. The idea was when you take a tumor, fresh tumor, you bring it to the lab. First of all, we sequence it very, very rapidly. So in less than 24 hours, we know what are the mutations are. And then we look and we consult the oncologist and say, which drugs should we test? So we take different drugs. Here are the biopsy. We slice it. We put into unique chambers in which we grow everything together, the cancer cells, the stromal cells, the bacteria, the fungi, everything that was originally there, whether we know it or not, we grow in these chambers. And then we can test multiple drugs on these slices of 3D slices of tumor, and we can just follow what happens to the tumor. When here you see one drug that kills all the cancer cells, and the other drug has almost no response. And then under 2 weeks, we can take these tumors, we can make blocks, we can make histology, we can consult pathologies, we grade and say how much killing was done by this specific drug on your specific tumor. This is a patient specific, and we report back to the patients to say, this is how much your tumor is sensitive to a specific drug. So need to stress, we don't really understand the mechanism. Sometimes we have two patients, they look exactly the same. Two colorectal cancer patients come to the clinic, right? And they have the same mutation profile, the same histology, the same everything. But you see that one patient responds well to drug A and the other to drug B. Why? If you give me a PhD in 5 years, I might be able to tell you the reason why. I don't know, maybe yes, maybe no, but the patients don't care about it. So what's nice about it that you can just immediately tell the patient if he's going to respond or not. So here you can see an example. That's how the tissue looks after a week in culture. So you see the cancer cells, you see stromal cells, they look exactly as the original tumor. And in this specific patient, when you treat with 5FU, you can see that there's no harm to the cancer cells. They're completely resistant. Why? I don't know. There's no good biomarkers today to 5FU. But if you give oxaliplatin or irinotecan, you can see that you kill the cancer cells. Again, I don't know why, but you would tell the patient you might not benefit very much from 5FU. This is just an example. And we made a clinical trial in Israel. There's many different hospitals in Israel that were part of it. And we found that there are very high sensitivity and specificity for our prediction. So just to give you a flavor, here's two patients very, very quickly. This is an ovarian cancer patient, came to us because she was after treatment and she progressed on treatment. So we took a biopsy, we sequenced it rapidly, and we found that she had an ERBB2 mutation, meaning that she might be responsive to a specific drug called neratinib. And her oncologist asked us to check different drugs. We checked all of these drugs, but neratinib actually had the highest killing score. So we found the mutation, and we showed that functionally, this patient should respond to neratinib, and she indeed got this treatment. Here's another that's a young patient with breast cancer, metastatic breast cancer, triple negative. And she progressed on therapy. And the plan for oncologists was to treat with Ribulin. But we checked the response of this specific patient to different drugs, and we found that her cancer cells were completely resistant to Ribulin, which made the oncologist change his decision to doxorubicin and to help the patient. So this is really a must stress. This is still, you know, still a novel technology that is not highly validated on thousands of patients, but we're working really hard to validate it as much as we can. For example, we started this year a clinical trial in London. It's sponsored by Imperial College, and we follow 250 patients in which we take a biopsy, we test it, we can tell after 2 weeks, predict if the patient is going to respond or not, and then we follow the patient for a few months and compare. Hopefully next year we'll have the results for all 250 patients. 2 last slides. I want to say that one can use these technology to look at novel drug combinations. Instead of testing them in vitro or in mice models, you can actually test them in human samples. We published a paper earlier this year showing this. I won't go into details right now. And my last slide is that you can also think about overcoming mechanism of resistance. So think about 2 patients that have the same mutation, 2 melanoma patients that have BRAF V600 mutation, right? And both are going to get a specific drug, say a BRAF inhibitor. But it might be the case that patient A would have some resistant mechanism X and patient B would have another resistant mechanism. Here we can test all the drugs to overcome all these resistant mechanisms and say to these patients, you know, you should get drug X and you should get drug Y to make you respond better to the therapy that you're going to get anyway, and we have a lot of work around it. So I started with this, that there's an urgent need for better precision therapy. I think what I try to present, there's 2 completely, utterly different directions. One is we really need to do good basic science. There's so much we don't understand in oncology. Even though we made so much progress, I feel like we're just in the beginning of an era. And I think, you know, these small studies, but others in the world, hopefully really trying to get more and more pieces of this unknown biology and teach us more. But at the same time, I think we need more and more techniques to help patients right now admit that there's a lot that we don't understand and try to overcome that by, for example, these functional screens and other ways that help patients benefit now from a better medical care. I want to say that everything I presented was done with extremely generous collaborators. There's so many, I cannot just present, of course, all their names, but I'm really thankful for them, for our funding mechanism. This is a part of the lab from a trip that we had a few months ago. And lastly, I want to thank my family that are actually here for supporting me along the way. So thank you very much, and thank you for listening.
So the disease is multifactorial.
Yeah.
But you're using only one drug. I mean, many things are involved. Exactly like in the neurodegenerative disease, there's a cascade of events going on. We know some of it, we don't know everything. So why not considering using a number of drugs that, especially in those people who are resistant to one drug? Right?
Yeah. So we don't use one drug. So in the oncology, as you know, there's a lot of combinations that are being used. If you refer to the last part about this technology, we usually test combinations of drugs and not a single drug. I'm sorry if my examples didn't portray that.
You use them at full strength or you use them at subluminal doses? Because we find in pharmacology, if you use a subluminal, there's a synergism at the lower doses.
So we try to get it. So it's actually there's a lot of it that went to understand what is the right dose to test ex vivo in order to predict human response to therapy. And that's, we can talk about for hours, maybe not for right now, but I want to say maybe there are 2 things. One, that in the tissue, we have also normal tissue, so we have cancer and normal. So we always make sure that the amount of drug that we add do not kill normal tissue, but rather the cancer cells. But also there's a lot of collaboration, years of collaboration that went into this process to understand what the right dose to use per each drug. There's different ways of doing it.
So do you think antibacterial and antifungal drug will be used in combination with
right. That's a great question also. I want to say like this, that antibiotics and antifungal are problematic because they affect not just the microbiome of the tumor, which I spoke about right now, but they affect our whole microbiome. And there is a lot of literature from recent years demonstrating, for example, that a good and healthy gut microbiome is really essential for a good response of patients to therapy, immunotherapies and other therapies. So giving antibiotics that will deplete the microbiome all over might not be the right way to go. So you can think of one option is to take an antibiotics directly to the tumor and affect only the tumor microbiome, right? That's one option. Another option is to deplete or affect the bacteria with different ways, phages or some other smart ways. And the third, which I like the most, would say, we don't need to kill the bacteria, we just need to change the activity of the bacteria. Maybe they have specific pathways that we want to do. And actually we have, right now, in my lab, we have a project in which we target specific enzymes in bacteria because we recognize that specific bacterial pathways are the one that mediate this immune resistance. So we don't try to kill the bacteria, but to change their metabolic function and take them from anti-immunotherapy to pro-immunotherapy. And we have, I think, exciting results demonstrating that this can be done. I think that would be a really interesting future direction.
I didn't get it. Is there any relationship, specific relationship between the types of bacteria and the normal intestinal microbiome?
So, no. So the gut microbiome, first of all, it differs along the gut. Right? So it's different microbiome in our stomach, in the small intestine, the large intestine. Mostly, I must say, the tumor microbiome is quite different from the gut microbiome. The maybe colorectal cancer has some similarity, but pancreatic cancer, for example, which also part of our GI tract, has a completely different microbiome from the gut, from the colon microbiome.
That means that this is not their origin.
We suspect that they actually come from the bloodstream mostly, and not from the colon. There is some iatrogenic, sometimes seeding. For example, patients that have ERCP, we see more bacteria that probably coming from the procedure. But I think that most of the bacteria are not coming directly from the gut, but maybe through the blood, and they can come from our mouth or from other places.
And the bacteria cause the cancer, or is it initiated? One of the things that you showed that the bacteria was inside the cancer. Was it inside the cancer before?
Yeah, it's a great question. So I would say like this: there are a few cancers that we all know that bacteria can initiate it. Maybe the prototype is the gastric cancer. We know that Helicobacter pylori presence in the stomach can cause different types of cancer. Viruses are known to cause cancers like HPV, right, and cervical cancer. The question is, are these other cancers, what fraction of the cancers are might be initiated by these bacteria? And the answer is we just don't know yet.
Very interesting. Now the new concept for Parkinson's disease, bacterial infection, starting in the gut. Yeah. Amazing story.
Yeah. Okay, thank you very much.
Thank you.
תודה רבה על ההרצאה המרתקת, ונעבור לזוכה הבא, נמרוד רפפורט, שהוא סטודנט מצטיין לתואר PhD במעבדה של פרופסור שמיר.
Wait. It's a different So hi, my name is Nimrod Ragpol. I'm a PhD student at Tel Aviv University. I'm a PhD student in computer science. So earlier you mentioned the AI in medicine, so I'm here to represent this research. I would like to thank the יודעים family, יודעים, and the whole family for this award. And I'll present to you, I'm from Tel Aviv University and the Weizmann Institute of Science. My advisors are Professor Ron Shamir and Professor Amos Tanai. And I'll present to you my research on multi-omic cancer subtype. So we know that cancer is heterogeneous, and it is a heterogeneous disease not only between tissues, but even within a specific tissue. And traditionally, therapeutic decisions were based mostly on the presence of pathological markers or single biomarkers, such as the presence of a single protein. But a couple of decades ago, experimental methods were developed that can measure not only one parameter per tumor, but tens of thousands and even hundreds of thousands. And each such kind of experiments that measure one kind of data is called an omic. So I'll give you some examples. You can measure now the whole genes that are being expressed in tumor, and this is one example of an omic. You can measure the DNA methylation of a tumor across pretty much genome-wide, which is another omics. And using these data, researchers have been, in the last couple of decades, trying to come up with definitions of cancer subtypes. Like specific subtypes of cancer that are different, that have biological difference, and hopefully also therapeutic differences. So we have been accumulating this omic data for 2 decades by now, but now we have data sets that only measure one omic per tumor, but they measure multiple omics per tumor. So for the same tumor, you can measure its gene expression and DNA methylation, microRNA expression, and so on. And the question is, can we, now that we have these multi-omic data sets, define cancer subtypes in a better way, in some sense, than when we had only one omic per tumor? So not only do we have these experimental methods, but there are actually publicly available data sets. So the best known of them is TCGA, which has more than 10,000 tumors from more than 30 different cancer types. They have multiple measure omics. So our goal is to define new cancer subtypes using the publicly available multi-omic data. So the first work that I did on this topic wasn't coming up with new algorithms, but just comparing existing algorithms. And before we can compare these algorithms, we should ask ourselves, how will we compare them? So we need some assessment criteria. So the problem here is that we don't have an accepted definition of what a tumor subtype is, because if we already had such a definition, we wouldn't need algorithms to come up with new definitions. So how do we, once we have a solution, how do we assess it? How do we say if it's a good one or a bad one? So we use 2 assessment criteria. One is differential survival. So we look at the cancer subtypes that we have, and we ask ourselves, is there a significant difference in survival between these subtypes? If there is, it means that there is at least some clinical and maybe biological relevance to these subtypes. So this one, and the second one is enrichment of known clinical parameters. I say here clinical parameters, but it can be things like age or sex, like is this subtypes enriched in a patient that are older? So it also can give some hint on the relevance of the subtype. Right, so what do we see here? We see the results of the benchmark, and this is only for data from glioblastoma, which is a brain tumor, and this is only using gene expression data, so only one omic. Every point here is a tumor, and the x-axis measures the differential survival, and the y-axis measures the enrichment of the clinical parameters. So you want to be as far to the right and as up high as possible. And this red vertical line measures the threshold for significant difference in survival. So if you're on the right of this line, you have a difference in survival. So when using only gene expression data, only one omic, only one algorithm found a solution which had a significant difference in survival. But when you use multi-omic data, so here it's gene expression, DNA methylation, and microRNA expression, you can see that almost all of the methods find a subtyping system with a significant difference in survival. So this example shows that at least in some cases, at least in glioblastoma, using more omics can, in some sense, help better define cancer subtypes. So now this is a bit of a diversion, but when we worked on this benchmark, like I said, we tested for differential survival between the cancer subtypes to see if they're any good. And the statistical test that we use is the log-rank test, is the standard test to test for significant difference in survival. But it has some issues that are known in the statistical community, but are less known for some of the people who actually use these methods eventually. So this test, there are many variations of this test, but the most widely used one is an approximate test, not an exact test. And what it means is that the p-value that this test reports is not the actual p-value of the result. And so I'll show you an example of an experiment we did that will make this more clear. We looked at breast cancer data, and we just randomly invented ways to partition them into subtypes. And for every partition, we used this log-rank test and looked what the p-value is. So by definition, because we just randomly created definitions of cancer subtypes, the distribution of the p-values should be uniform. But what you see here, that if we use the threshold of 0.05 to say that the subtyping has significant difference in survival, then actually 0.1 of the subtypes will have will appear to be significantly different. So, this approximate test doesn't control for the false positive rate, and we implemented a different version of the test that does control the false positive rate, and whenever we did the analysis, we used this correct version. And also on the paper that we wrote on this research, we identified some previous features that falsely called differential survival. Right, so back to cancer subtyping. So after the comparison I did between different methods, I developed 2 methods to do multi-omic cancer subtyping. So one of them is NEMO, and it's kind of an acronym for neighborhood-based multi-omics clustering. And here you see the results. Here there are aggregated results across 10 different cancer types, and every point is an algorithm. You can see NEMO here, and you have the number of enriched clinical parameters and the differential survival, again, across 10 different tumors. And you see that NEMO, it's not the best in either of them, but it's second in both of these assessment criteria, so overall good results. It is very fast additionally, so it takes about 10 seconds to run across hundreds of patients. And it's the only algorithm that knows how to handle partial data. So what is partial data? In some of the publicly available data sets, you will have, for some of the patients, only a subset of the omics that are being measured. So you can have some patients that have gene expression and DNA methylation, but another subset of them will only have gene expression, another subset will only have DNA methylation. So NEMO is the only algorithm that can also detect cancer subtypes using such kind of data. Okay, so NEMO works under the assumption that there is a common structure to the different omics. What does that mean? That if two samples are similar, two tumors are similar in one omic, so in gene expression, they will also tend to be similar in the other omic, in DNA methylation, for example. And this similarity can be weak, but because we're using multiple omics, this possibly weak signal will become stronger. But this assumption doesn't always hold, and I'll show you an example. Here what we see are T-SNP plots, and I'll explain this one first. So every point here is a tumor, and two tumors that are close to one another in their gene expression profile are closer to one another in this panel. And here is the same for DNA methylation and microRNA expression. And each tumor has the same color in all the three different panels. And this is data for breast cancer. So here we see all of these purple tumors, and you see that they're similar in terms of their gene expression profile and methylation profile and microRNA profile. But if you look at these yellow tumors, you see that they are similar in terms of their microRNA profile, but they're not very similar in gene expression, and they're not similar at all in methylation. So what that means is that tumors that are similar in one omic are not necessarily similar in another. And I developed an algorithm that detects cancer subtypes using this assumption. So it's called Monet, and instead of finding cancer subtypes, it finds what we call patient modules. So I'll show you what are these patient modules. So here we have a schematic figure. These black points are the tumors, and these three patients, they belong to the same module, to module alpha, and these three patients belong to the same module, to module beta. And you see that there's a circle encircling these patients. And what the color of this circle means is that the patients that are part of this module should only be similar to one another in terms of the surrounding omics. So these three patients are only similar to one another in this red omic and in the green omic, and these three patients are only similar to one another in the blue omic. So here you can see that patients that are part of the same module don't have to be similar to one another in all omics, but only a subset of them. Okay, I won't describe to you the algorithm in interest of time, but I'll show you the results. So we applied this algorithm to 10 different cancer types in TCGA, and we used the three omics that I mentioned previously: gene expression, DNA methylation, and microRNA expression. And first, we wanted to see, do we actually see the phenomenon that I mentioned, that we find patient module that only that patients in these modules are only similar in the subset of the omics. So this is what you see in this Venn diagram here. And indeed, we found such modules. So for example, we found 15 different modules across the 10 different cancer types who had patients that were only similar in terms of the gene expression profile. But we also found, let's say, 8 modules that were similar both in terms of methylation and microRNA expression. We found 6 modules that are with patients that are similar across all omics. We next compare our algorithm to other algorithms. So this is again the assessment criteria that I mentioned in the beginning of the talk. So this is the number of cancer types in which you found a significant difference in survival. This is the number of cancer types in which you found an enrichment of known clinical parameters. And Monet is the pink bar, which has the best performance in both of these assessment criteria. It is tied together with NEMO, which is the first algorithm that I developed. Maybe it's not so surprising, but NEMO and Monet had good performance on different cancer types. So we think that these two methods can be used complementarily. We next zoomed in on Monet's results on ovarian cancer. So Monet found 4 different patient modules on ovarian cancer, and they all covered the gene expression only, meaning that in all modules, patients were similar to one another in terms of their gene expression profile, but only module number one of the modules, covered the microRNA omic, that is, patients in this module were similar in terms of their microRNA expression profile. Monet found a significant difference in survival between the modules that it detected, and it is the only algorithm that we used found such a difference in survival. Okay, there were differentially expressed genes between the different modules, especially the immune response and the expression of genes relating to the extracellular matrix. And now back to this module 1, I mentioned that it has samples that were similar in terms of the microRNA expression profile. So we asked ourselves, what are the microRNA that these patients have in common that is different from the other patients? So this is what you see here. Every point is a microRNA, and you see its expression in module 1, and you see its expression in the other patients, and there are some overexpressed microRNAs. One of them is microRNA 514, which was recently reported to have a role in cisplatin resistance in ovarian cancer. So this is a recapitulation of a previously reported result. And there are additional microRNAs here that are upregulated and previously weren't reported. Right, so to summarize, I talked about multi-omic subtyping, how to find cancer subtypes using multi-omic rather than single-omic data. I mentioned the problem with the commonly used log rank test, at least its most widely used variation, which doesn't control the false positive rate. And I mentioned that we developed a method that does control for the false positive rate. Mentioned NEMO, which performs multi-omic cancer subtypings and assume that there is some common structure between the omics, and then Monet, which doesn't use this assumption and allows samples in a cancer module to only be similar in a subset of the omics. All of the code for all of these methods is publicly available, so everyone who wants to use them can use them. I would like to thank Roy Safra, with whom I worked on Monet, and my advisors Ron Shamir and Amos Tanai. To thank you for listening, the funding agencies, to thank you for listening, and again, thank you for the יודעים family for this award.
Wow. הזוכה השני, בלבד גונדירש, מהמעבדה של פרופסור צ'חנובר, בחקר הרפואה בטכניון.
לא, עשיתי משהו לא טוב.
So hi everybody. First I would like to thank Professor יודעים and the יודעים family, the selection committee and the organizers for this wonderful meeting. Thank you for the opportunity to be here today. It's really a great pleasure and honor for me. I'm going to talk about two projects that I had the opportunity to be involved during my PhD studies. Both of them deal with unraveling the tumor suppressive mechanisms by the ubiquitin ligase KPC1 and the NF-κB protein P50. So let's start. Ubiquitination is an enzymatic cascade that leads to the transfer of the ubiquitin moiety to the substrate protein. This ubiquitin transfer is mediated by the E3 ubiquitin ligases. The ubiquitin ligases are the specific arm of the ubiquitin system. There are about 1,000 different E3 ligases, and each one recognizes specific proteins for ubiquitination. The basic dogma is that this process repeats a few times, and then the ubiquitinated substrate undergoes degradation in the proteasome. Of course, there is much more than the basic dogma. There are many types of ubiquitinations and many fates to the ubiquitinated proteins. The subset which we have been working on belongs to the NF-κB family. NF-κB proteins are strongly correlated to tumor progression. Many tumor types consistently express NF-κB proteins. They regulate genes that contribute to metastasis, oncogenesis, inhibition of apoptosis, and many more. Following ubiquitination, P105, the NF-κB protein that we have been working on, can either degrade completely or process to the active subunit P50. A few years ago, a lab identified the E3 ubiquitin ligase KPC1 as the ubiquitin ligase that mediates the ubiquitination and proteasomal processing of P105 to P50. One of the most interesting parts of this work is the finding that overexpression of KPC1 or P50 in xenograft in mice has a strong tumor suppressive effect. So during my PhD, we have been working on two aspects of the tumor suppression mediated by P50. We focused on continuing unraveling the tumor suppressive characteristics of KPC1 and P50, and also how to generate an access of P50 by stimulating the processing of P105 to P50. So to continue investigate the tumor suppressive characteristics of P50, we've done mass spectrometry analysis on tumors that overexpress P50 and compare them to control tumors. You can see dramatic suppression in PD-L1 expression in tumors or cells that overexpress KPC1 or P50. The control tumors have 4 to 5 times higher expression of PD-L1 compared to the P50 tumors. And in immunohistochemical section of the tumors, we see a massive infiltration of leukocytes into KPC1 and P50 tumors. So we asked whether the leukocytes infiltration contributes to the tumor suppression mediated by P50. We compared 3 mice types, actinic nude mice which lack T cells, skid mice which lack T and B cells, and NSG mice which lack T, B, and K cells and macrophages. We can see that there is no significant difference between the nude and skid mice. We can see it also in the CD45 staining. Both the P50 tumors are overflowing with leukocytes. So we concluded that B cells don't play a role in this tumor suppression. However, the difference between nude mice and NSG mice is significant. Therefore, the leukocytes infiltration does play a role in the tumor suppression mediated by P50. Moreover, NK cells and macrophages are the leukocytes that infiltrate the P50 tumors and leads to the suppression. So to further confirm that indeed the NK cells and macrophages infiltrate the tumor, the P50 tumor and inhibit their growth, we conducted adaptive transfer experiments. We constituted the NK and macrophages population in NSG mice, and we can see that due to that NK cells and macrophages selectively suppress the growth of P50 tumors and doesn't significantly affect the control tumors. So I wanted to check which chemoattractants P50 regulates and supposedly attract the NK cells and macrophages to the tumors. So we did cytokine array, and we can see that due to the expression of P50, the expression of CCL3, 4, and 5 is elevated. So we express each of them in UAT7 cells, and we can see leukocytes infiltration and dramatic tumor suppression. Here, it's a bit slow. So this is a summary of our findings. When KPC1 is low, the amount of P50 is low as well. We assume that in this case, P50 dimerized with P65, and PD-L1 can be transcribed. When KPC1 is high, it triggers the processing of P105 to P50. We assume that in this case, P50 dimerized with another P50 and create P50 homodimer. This homodimer migrates to the nucleus to regulate gene transcription. It inhibits the transcription of PD-L1 and increases the transcription of CCL3, 4, and 5, which lead to activation and recruitment and activation of macrophages and NK cells. The second project started with the goal to characterize the interaction site of KPC1 to P50, and it drifted into finding ways to force the processing, or stimulate the processing of P105 to P50. So to make long story short, we found a sequence of 7 amino acids that is necessary for the binding to P105. The presence of this sequence forces the interaction with P105 regardless to the location in the protein, and the deletion of this only 7 amino acids eliminate the interaction of the wild type KPC1 with P105. So we wanted to check whether small mutants of KPC1 that contains the 7 amino acid binding site can suppress tumor growth. You can see that the small mutant of KPC1 that contains the 7 amino acids has strong tumor suppressive effect. And as we found in a previous paper that KPC1 and P50 attract leukocytes into the tumors, you can see that the small mutant that contains the binding site attracts leukocyte as well. I don't have enough time to go into the mass spectrometry analysis of the tumors, but we can see that by the proteomics analysis revealed that both the tumors that overexpress KPC1 or the small mutant that contains the 7 amino acid binding site are pretty much the same. So based on these findings, we generated PROTAC molecules. Generally, PROTACs are chemical complexes made of 3 parts, a binding ligand for the target protein, an E3 ligase ligand, and linker between. PROTAC molecules form a ternary complex with the E3 ligase and the target protein, thus forcing the ubiquitination of the target protein by the recruited E3 ligase. Sorry. Nowadays, the PROTAC method to inhibit and degrade the oncogenic proteins plays an important role in the development of therapies for many malignancies. Some of them are in clinical trials. So we generated PROTAC consisting on both the VHA ligand, which bind the VHA ubiquitin ligase, and the 7 amino acid base peptide that we found is necessary for the interaction with the P105. You can see that PROTACs that contain the 7 amino acid binding site are able to force the ubiquitination of P105 by the recruited VHA ligase. Lastly, we wanted to check the ability of the PROTAC to stimulate the processing of P105 to P50 in cells. We can see that without the PROTAC, the interaction of VHL with P105 is negligible, and in the PROTAC forces the interaction of P105 with VHL. Importantly, adding the PROTAC forces the processing of P105 to the active tumor suppressor P50. We can see that in the presence of the PROTAC, P50 accumulates. So I would like to thank especially to Professor Chechanover and Dr. Elena Kravtsova for basically everything. We've been working on this project, both projects together for the last 6 years, and to all the lab members and collaborators. Thank you.
What's the mechanism PROTAC activating P450?
The activation of P50 is by processing of the precursor P105 to the active subunit P50, which is originally or natively mediated by the KPC1 ubiquitin ligase. The PROTAC forces the ubiquitination of P105 by the recruited VHL ligase. So this is how it's successful. It's successful in vitro and in cells. The PROTACs that we have made is based on a sequence of 7 amino acids, and
you made different derivatives.
We built different derivatives. Some of them are more potent, some of them are less, but we are still working on improvement and adjustment of the part that binds the P105 in order to make it more potent and hopefully works in vivo also.
תודה רבה גם לגלעד, ותודה רבה לפרופסור יודעים על מסורת הפרס, ולכולכם על ההשתתפות. ברכות חמות לזוכים, ונתראה בכינוס הבא.